So, hi, good evening. My name is Nandika. I'm a PhD student from IIC Bangalore, and welcome to this problem solving session for experimental biotechnology. Today, we'll be discussing content related to week 11, where we'll discuss various sequencing methods as well as the concepts of northern and southern blots. Okay, let's get started. So, as we already know, and we revised last week, DNA is made up of 4 nucleotides. We have the pyrimidine nucleotides, basically thymine and cytosine. In the nucleotide form, we'll refer to them as deoxythymidine monophosphate. This is DTMP, and deoxycytidine monophosphate, right? So basically, the sequence is determined by which nitrogenous base is attached over here. So again, depending on the base, we have pyrimidine nucleotides and purine nucleotides. In the purine nucleotides, we have adenosine and guanosine, or rather deoxyadenosine monophosphate, deoxyguanosine monophosphate. And DNA is made up of a polymer of these different nucleotides. Right? So again, as we saw last week, DNA has 2 strands of opposite polarity which wrap around each other to form this double helix. And information is encoded by the sequence of these nitrogenous bases within the DNA. Right? We also saw that the complementarity of these 2 strands is what allows for DNA replication and so on, and ensures the fidelity of information transfer. But the information itself is encoded within the sequence of these nitrogenous bases. So ATGAC is the part of this polymer which is actually encoding the protein sequence that will be made out of it. Right? And so if we wanted to determine the sequence of the bases within this polymer, within this DNA molecule, we would need to read what each base is one by one. Right? So there are a number of different ways to do it, a number of different methods of so-called DNA sequencing, and we'll discuss a couple of them today. The first is what is called first generation sequencing or Maxim-Gilbert sequencing. So this was proposed in around the mid-70s, where basically this is based on a series of 4 different reactions that will modify the DNA at different points. So one reaction involves incubating the DNA with formic acid. Okay, and what formic acid does, it will result in depurination, which is loss of the purine base. So this will modify the DNA wherever purine base is present. So that would be at adenine and guanosine. Right? So at A and G, we would get a modification when we react it with formic acid. However, if we react the DNA only with dimethyl sulfate, okay, dimethyl sulfate will cause methylation only at guanine. So only at G. So you can imagine that if our DNA sequence had an A, then A would react in this reaction, but it would not react in the second reaction. Whereas G would react in both reactions. So this is where we can tell the difference between the 2 purine bases. Similarly, for the 2 pyrimidine bases, we have 2 different reactions. The first is to react it with hydrazine. This will cause hydrolysis at both pyrimidine bases, that is at both C and T. The specific reaction is caused by adding salt in this previous reaction. When we add salt, that blocks hydrolysis at thymine, and we only get hydrolysis at cytosine bases. Okay. So what these 4 reactions are doing are modifying the DNA at various points. This modified DNA is then cleaved by hot piperidine. So it is cleaved at the phosphodiester bond after the modification. And so the workflow for Maxim-Gilbert sequencing reaction would be as such. So first, we'd have to label the DNA fragment. Okay, single-stranded DNA labeled at one end. So you can label it either 5' or 3', but here they've labeled at the 3' end. they've added a radioactive phosphorus molecule, and this is so that we can detect the different DNA fragments later. Now, here they've discussed one case, which is the cytosine reaction. Okay? So, this will be when we incubate the DNA with hydrazine and salt. As a result, we'll get modification at cytosine only. So, if you look at this DNA sequence, there are 3 C bases, right? 1, 2, and 3. And the ratio of the reaction mixture is taken such that on the balance of probability, each DNA molecule will only be modified once, and each site is equally likely to be modified. Okay? And if under these two assumptions, when this molecule is treated with the C specific reaction, we should get one set of molecules which is modified at this C, one set of molecules which is modified at this C, and one set of molecules which is modified at this C. Okay? Now remember, now that we have modified the DNA, we are then going to cleave it using piperidine, and piperidine will cleave the phosphodiester bond after the modified base. Right? So if this C is modified, the DNA molecule will be cleaved here, in between the C and the G. And what we are going to detect is the leftover fragment after cleavage. Since we have labeled this 3' end, we are going to detect whatever is on the 3' end after the modified base. So if this C is modified, then we've cleaved it with piperidine. The fragment we are going to be left with is just this single G residue. Right? Similarly, once this C is modified, we will cleave here and we'll get the sequence TCG. If this C is modified, cleaved here, we will get the sequence GCT TCG. Right? So after cleavage, we will end up with this set of labeled fragments. We'll also have these other fragments left, but they are non-radioactive, and so we will not be able to detect them in our later stages. Now you can see that all of these fragments are of different sizes. So we can then separate them on the basis of their size through electrophoresis. Right? So we will run them on an agarose gel. We call it a sequencing gel because it's going to help us determine the sequence. And then we are going to do autoradiography to detect the radioactive fragments. Now this is just one of the 4 reactions. This is the reaction that will give us the position of C's in the sequence. If we run all 4 reactions side by side, what it will give us is the position of all 4 of the products. Right? So the reactions we would have would be A plus G, G, T plus C, and C. And then we will run a gel with using fragments from all 4 of these reactions. So here we can see from the only C reaction, we're going to get these 3 fragments. So since this is the smallest fragment, it will run the furthest. So you get a band here. This is slightly bigger, we'll get a band here. This is even bigger, so we'll get a band here. Okay. Now in the T plus C reaction, we're going to get bands corresponding to both of these pyrimidine nucleotides. So in the T plus C reaction, we will get this band over here. Okay. You'll notice there's a thymine over here, right before the cytosine. So in the T plus C reaction, we will also get this product. Right? Since it's being cleaved after this thymine. So we'll get another band here. Right? We get a band here. Get a band here. And here. So now here, by comparing these two, if the band is present in both of these, we know that the sequence must contain cytosine. However, if the band is present only in this lane, but not here, it must correspond to a T. Right? Similarly, if we look at the purine reactions, this will cleave after this G, so we will not get any product for this, right? You won't be able to sequence this last residue. But what we will get is bands for these other products. Okay, so there'll be one G, right? Let's give us bands in both. Then we have an A over here, right? And since there's a band in both of these, we know that this must be a G. And since there's only a band here but no band here, this must be an A, right? Since we have labeled the 3' fragment, we will have to read the DNA sequence like this from 5' to 3', right? So the DNA sequence is going to go like this. Okay? And this is essentially how we would read a sequencing gel in the Maxim-Gilbert method. So this works very well for short stretches of DNA, right? You can see this was a sequence of 8 amino acids, and this can be easily deciphered using this method. However, it's also not the most efficient method, since you have to do these multiple reactions, and it was soon overtaken in popularity by a method we still use today called Sanger sequencing. It's also called chain termination. Chain termination or dideoxy sequencing method. What Sanger sequencing does, named after the inventor Frederick Sanger, is essentially replicate the process of DNA replication, except that it incorporates something called a chain terminator, okay, which is this dideoxynucleotide. Okay? So this is a regular nucleotide. You can see deoxyribonucleic acid, which means that we don't have an OH in the 2' position. However, this OH in the 3' position is what is used to form the phosphodiester bond with the next residue. Right? So we know the 3' OH is required for chain elongation. This is what a normal DNTP would look like. However, if we were to replace this OH with an H, right? Now this is a dideoxy, 2' and 3' dideoxyribonucleoside. And since it's lacking this 3' OH, it cannot form a phosphodiester bond with the next residue, right? As a result of which, the chain cannot continue after this has been incorporated. Okay? This will be added to the chain like any other nucleotide because it has a phosphate group. But after this is added, no other phosphate group can form a bond with this. So this will automatically stop the elongation of the chain at that position. And in Sanger sequencing, one of these chain terminator precursors will be included to stop the elongation process at random points. And what we are basically going to do is run a gel of these randomly stopped reactions to get an idea of the sequence. Now, since we are replicating the DNA replication process, we do need everything that is required for DNA replication. As we discussed, you'll remember, for DNA replication, we need a template. So this is your DNA to be sequenced. We know that DNA polymerase cannot start adding fragments de novo. It requires a primer. And similarly, in Sanger sequencing, you will require a primer. It is complementary to the starting of your DNA sequence. We will need all the precursors, right? DATP, DCTP, DGTP, and DTTP. Okay. In traditional Sanger sequencing, these will be radioactive. Okay. And that is so that we can detect the fragments later. Same deal with Maxim-Gilbert, we're labeling that at one end. Sanger sequencing, we'll use radioactive nucleotides so that we can detect our products at the end. And of course, we need a polymerase. So the polymerase used in Sanger sequencing is something called the clinofragment. This is the large subunit of DNA pol 1. Okay. What this has is the extension capabilities, that is the polymerization capabilities of DNA polymerase 1. It is just lacking the 3' to 5' exonuclease activity. Okay. And this ensures that all of our products which are formed by Sanger sequencing have the same 5' sequence. Okay. So we have a reaction mixture which has a template DNA. We have to add a primer. So you can see this is our template DNA. We have a primer which is going to be complementary to this part, okay, which is basically the starting of the sequence or towards the 5' end. Right? Or sorry, so this since it's going to be synthesized in this direction, the primer has to bind here. So this is going to be complementary to the sequence here and is going to bind over here. Okay, we're going to add our nucleotides, and then we're going to split the reaction into 4 parts to add in each reaction one of the chain terminator nucleotides. So we will have one reaction which has DD-ATP, one with DD-CTP, one with DD-GTP, and one with DD-TTP. Right? So in this reaction is going to give us the position of every A. This reaction will give us the position of every C. This reaction will give us the position of every G. This reaction will give us the position of every T. So we will split it into 4 reactions, add DNA polymerase, and allow it to polymerize. Now you can see as this fragment is formed, let us consider this case where we've added DD-ATP. This is going to be extended in this direction until it reaches one of the adenine residues. And here, randomly in some cases, DD-ATP will be incorporated instead of DATP. And that will stop the chain elongation at that point. And so we will get a product that is this big. Similarly, for the other adenine residue, we'll get another product which is the size of that DNA stretch. Okay, so this is going to give us 2 different products. Similarly, for each of these other reactions, we are going to get different product sizes corresponding to the position of that residue in the sequence. Now, similar to Maxwell-Gilbert sequencing, we're now going to separate all these fragments on the basis of size and then look at the sequence using autoradiography. So now the smallest fragment would be, if this is the primer, we just add another G, right? So that would migrate all the way to the end. That will be down here. Okay. Yeah. And so with increasing size, right, fragments will appear further and further up. And to read the sequence, we can now start from the end. See, there's a C here, C, A, C, G, T, A, T, G, right? Which is the 3' to 5' sequence of the complementary strand which has been synthesized during the Sanger sequencing process. Okay. That is how you read a Sanger sequencing gel. So yeah, here's what a real life example will look like. We run the 4 reactions which contain the 4 different chain terminator DNTPs. And depending on the position of the band, we can then read the DNA sequence from 5' to 3'. Okay. Now, you can see that autoradiography is not the best technique for this, right? Since you have to work with radioactive material and so on. And it has now been automated into a fluorescent method wherein we add a fluorescent tag to each of the chain terminators. Okay? And so we have now eliminated the need for having 4 different reactions. You can add the 4 different chain terminator DDNTPs with 4 different fluorophores and then just detect the sequence of the fluorophores in the gel. Right? So how that would work is set up a DNA polymerization reaction containing the template strand, primer, the DNTPs, DNA polymerase, and all 4 DDNTPs, each labeled with a different fluorescent dye. Right? So here you can see they're shown in 4 different colors because each of them has a separate fluorophore. Then once we allow this reaction to take place, we will get synthesized chains of different lengths in which each DDNTP, a chain terminator, has been incorporated at its corresponding position. Right? And so we can just load all of these products on the gel and detect the sequence of the fluorophores in the gel. Right? So if T is blue, right, I can tell that this has to be a TT A blah blah blah. Right? So I can just read off the sequence of these products. Okay? So we record the sequence with scanning laser fluorescence detector and computer. Right? You can do this by capillary electrophoresis. Each of them will pass through a thin capillary in front of a laser, which will detect the fluorescence and read like this. And so the readout you would get from this Sanger sequencing would look something like this. We'll get a peak corresponding to each fluorophore. Okay? And if each of these peaks is well resolved, okay, and symmetrical, then you will know that this is a good sequencing result, okay, a clean sequencing result. Like here you can see we have a small amount of red fluorescence, but the green fluorescence is greatly outweighing that, which is how we know that this green fluorescence is the actual read for this position. Okay. And Sanger sequencing is very useful for sequencing sort of mid-sized DNA molecules around 1 kb or so. You can get fairly good sequencing outputs from this method. Sanger sequencing is now not used as much anymore as we have moved on to more advanced and more high throughput methods of sequencing. So here you can see how it compares to some other methods. So this is something called pyrosequencing, where essentially the incorporation of each new amino acid emits a small flash of light, and it's the flash of light which is detected as the readout. Okay, this is done by incorporating a special kind of pyrophosphate and luciferase, which converts the energy of this addition into a small flash of light. Okay. This is by far the most well-known sequencing method for large amounts of DNA, something called Illumina, okay, which is called reversible terminator technology, where essentially here the DDNTP irreversibly binds at the end of the DNA sequence, right? So once this is bound, there is no way that we can remove this and then continue extension. But how Illumina works is that they have a terminator which reversibly binds. Okay, so the terminator binds so that for some time elongation is halted at that spot. A read is taken for the terminator. It is then removed and elongation is allowed to continue so that we can read further spots on the same DNA sequence. Okay, and it is conducted on a chip where each of these sort of colonies is giving you reads. Right, so you can see this is massively parallel DNA sequencing. Okay, where we can handle very large numbers of samples. Now, all of these methods are good for sort of mid scale DNA sequencing. If you want to get extremely long reads, right, then we have to go for other types of sequencing methods, such as Oxford Nanopore or so on, which will give you millions of base pairs of reads. Okay, so depending on your application, there is now probably a sequencing method for it. Okay, but in a lab today, if you want to sequence, say, a small part of your gene and send it off to a sequencing company, they will probably be doing Sanger sequencing on your gene because it's easy to perform and it's very reliable. Okay. So now that we've understood the basics of sequencing, DNA sequencing, we can go ahead and look through some questions. Okay, here we go. For analyzing DNA sequences, two techniques have been developed. One is a DASH method, frequently termed DASH, involving enzymatic synthesis of DNA and stopping the synthesis of specific nucleotides. Right? Enzymatic synthesis of DNA occurs in Sanger sequencing, as we discussed. Right? So it's an enzymatic method termed as Sanger sequencing. While another technique, the DASH method, is often used for sequencing for small fragments of DNA. As we discussed, Maxim-Gilbert method is good for sequencing very small fragments. So the chemical cleavage method of DNA sequencing, developed by Maxim and Gilbert, is often used for sequencing small fragments. This answer fills all the blanks. Which of the following are advantages of using capillary electrophoresis in Sanger for detection of sequenced DNA? Right, so why should we use capillary electrophoresis instead of doing the standard long agarose gel and then doing autoradiography with it? Right. So thin capillary tubes, they allow us to avoid using sequencing gel and requires little manual operations. It allows it to be automated. It's correct. The electrophoretic runtime is longer. This is also correct, but it's not an advantage. A high throughput can be achieved. So the two advantages of this are 1 and 3. In experiment, which of these are not required for the preparation of reaction mixtures? Okay, so which of these should you be avoiding when looking to sequence your DNA? Now, one, if you remember from last week, we discussed that divalent cations such as magnesium are essential for the functioning of polymerases, DNA polymerases. So what you don't want to do is chelate your divalent cations, right? Because that is going to stop your polymerase from functioning and you will not get any result for your sequencing reactions. And so one thing you should definitely avoid when you're doing any enzymatic reaction related to DNA polymerase, you should avoid the addition of EDTA. Okay. What is the difference between a deoxyribonucleotide and a dideoxyribonucleotide? So we already know that a deoxyribonucleotide is missing its 2' OH, whereas the dideoxyribonucleotide is also missing the 3' OH on sugar. Right? So the dideoxynucleotide is missing the 3' OH group on its sugar. Otherwise, the nucleotides are functionally identical. Okay? So they both have phosphate group, the nitrogen base, and so on. It's just the matter of one OH group. Okay, starting from the sequencing primer, what is the sequence of this DNA sample? Okay, so remember, it will be sequenced 5' to 3'. Okay, so let's try to read this sequence. So G, T, A, C, 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 G, A, T, C, A, G, G, A. Okay, so we've just read the sequence of this. Okay, but if you look at the question, it says starting from the sequencing primer. Now, where will the sequencing primer bind in this reaction? It will bind at the 5' end, because DNA polymerization will start in the 5' to 3' direction. So it requires a primer to be upstream of where we want to sequence the region. So the DNA primer will be here prior to this G. So our sequence from the DNA primer will start with the G. Okay, so now we need to look at which of these matches our DNA sequence. So of these two options, we need to choose GTACCC, seems to be true for both, GAAA, GAAA, TCA, GGA. This seems to match the sequence that we've got from here. So this is the correct option. In the Sanger sequencing technique, given this template strand, which of the following could be used as a DNA primer? Remember the template strand they have given in the 5' to 3' direction. But DNA synthesis on this strand has to go in the 3' to 5' direction. Right? It will start from this G and be extended in this direction, because this will be 5' to 3'. Okay, for our newly synthesized strand, which means the primer has to bind to the 3' region of this template strand. So we should get a primer which is complementary here. So the sequence should be C, C, T, T, G, T. Is there something which vaguely matches this? Yes. C, C, T, T, G, T, A, C, T. Yes, this seems to match this sequence up to about here, right? Which is sufficient for its use as a DNA primer. And so we will go ahead with this as the only option that can be used as a DNA primer for Sanger sequencing. Okay. If we have DATP, DCTP, DDCTP, and DDGTP, okay, so both C and G chain terminators are there in this reaction. Which of the following strands could be produced from a sample containing this template and primer? Okay, where is this primer going to bind on this template? So C, A, G, T, G, G, A, T, A, T, C, T, G, T, T. Okay, so primer is going to end over here. And then the rest of the strand is going to be extended from the primer. Okay. Now, in this extension, since both C and G chain terminators are there, it can theoretically be terminated at any of these points. Okay, TT. So this would be extended to A, C, A, G. Okay, so this is possible. Right? It's been extended to this point. GCC, this is also possible. GAA. So here it's been extended to this point, but there is no A chain terminator in this reaction, right? Which means that there is no way for the synthesis to stop at this A residue. It would have to go to this next C and then stop. So this will not be produced in this reaction mixture. This is terminating at A, not G or C. So within this mixture, A and B are both possible, since they're terminating with either the DDCTP or the DDGTP. In DNA sequencing, which enzyme replicates the DNA? This is DNA polymerase. what is the function of the template DNA during DNA sequencing? The template DNA is obviously providing the DNA sequence that we wish to determine, right? So the answer here is B. Okay, what type of DNA cleavage is done in the Maxim-Gilbert method? So Maxim-Gilbert method, DNA cleavage is done so at modified bases. So it's not cleaved at the edge. It's not cleaved in a gene-specific manner. It's not cleaved in a base-specific manner. It's cleaved in the middle of the DNA, right after a modified base. Which of the following is not required for DNA sequencing? So PCR helps to amplify the DNA template. Also in the case of Sanger sequencing, single-stranded DNA is required, right? Since we need it to act as a template for DNA polymerization. So PCR is used to amplify the DNA template for DNA sequencing. Cloning. So since single-stranded DNA is required, sometimes it will be cloned into an M13 phage, which will give you a single-stranded template, but it's not a requirement as such. Electrophoresis is obviously required to separate the different products of the sequencing reactions, and restriction digestion is often used to break the DNA into smaller fragments so that the fragments can be sequenced individually. Okay, so of these, what is not required specifically for DNA sequencing is cloning. Okay, any DNA fragment can be sequenced. N-labeled DNA sequencing is known as dideoxy method. This is false. We already saw that the DNA sequencing method in which the end of the fragment is labeled is the Maxim-Gilbert method, since we're labeling one end of the DNA fragment with a radioactive nucleotide. In Sanger sequencing, it is labeled throughout by the incorporation of radionucleotides. The 32P is added at the 3' end by polynucleotidyl kinase. This is false. Polynucleotidyl kinase will add 32P to the 5' end, okay, of the DNA reaction. The N-labeled fragment is cleaved into how many pieces? It's asking, basically, this is our fragment. We have labeled it here. How many pieces are going to be cleaved into? So each fragment, we assume the probability of labeling is 1, okay, which means that the probability of modification is that it will be modified at one site only. So it will most likely be cleaved into 2 pieces, right after the label site. Restriction digestion is the only process to achieve sequencing by the Maxim and Gilbert method. This is false. You can also do PCR to generate a large amount of templates, which can be used for Maxim-Gilbert methods. Okay. The denatured strands cannot be separated. This is false. So for Sanger sequencing, usually you will denature the DNA before sequencing because you need a single-stranded template. But obviously, the denatured strands can be separated because that is the point of the denaturation process. Okay. What are the base-specific cleavage sites used in the Maxim-Gilbert method? So it's basically asking what are the 4 reactions. So we have the 2 purine reactions, A plus G and G, and the 2 pyrimidine reactions, C plus C and C. This is the 4th answer. G, C, A plus G, C plus D. Okay, which step is not involved in base-specific cleavage of DNA fragment? So modification of concerned base is required. Removal of modified base from DNA strand, induction of random strand break, and label variable lengths produced. Right, so the DNA strand is not removed, right, from the DNA fragment. We just cleave the DNA fragment after the modified base. So this is not involved in base-specific cleavage of the DNA fragment. The dideoxy method is also known as Sanger's enzymatic sequencing, right? Because in Sanger sequencing, we use the dideoxy nucleotides as chain terminators. Which enzyme is used in replication in Sanger method? As we discussed earlier, we use polymerase 1, but we don't use the full polymerase 1. We use the clinofragment, which is the larger subunit of polymerase 1. With respect to Sanger's enzymatic method, pick the odd one out. Primers we require, clinofragment we require, restriction digestion is not essential for Sanger's enzymatic method, so this is the odd one out. DDNTP act as chain radioactive markers. This is false. Where the DDNTP just acts as the end marker. It does not necessarily have to be radioactive. Strand separation for DNA sequencing is done to sort fragments according to their size. Okay. Which is how we can then read the sequence back. The position of the band during electrophoresis indicates that the DNTP has been used as indicates the DNTP that used as a chain terminator in the mixture. Now, the position of the band does indicate the chain terminator that is used, but the DNTP does not act as a chain terminator. Remember, the chain terminator is a dideoxy nucleotide phosphate or a DDNTP. So the DNTP has no role in chain termination. This is false. Okay, which of the following is not a DNA sequencing method? So pyrosequencing, Maxim Gilbert, and chain termination we discussed. Edmund degradation is a protein sequencing method that we will discuss in the following slides. Okay, in Sanger method, the DNA fragment of DNA should first be dash. So it first be PCR amplified before proceeding for sequencing. Okay. Sanger's method is based on the principle that dash the DDNTPs stop PCR amplification. It stops extension of that, which is how we can differentiate different fragments based on the size. Okay, DDNTPs differ from DNTPs in that they lack OH group from the 3' carbon. How many primers are used in Sanger sequencing? Right, so we only need the one primer, which is going to bind to the 5' end of the newly synthesized strand to allow DNA synthesis to begin. So we need just one primer. In modern automated DNA sequencing, the modified Sanger method is used where we have DDNTPs with different fluorescent markers, so right, which allows us to run a single reaction and then read out the method through capillary electrophoresis. Which of the following does not occur in Maxim Gilbert method? Right, so in Maxim Gilbert method, there is no G plus C reaction. Right? All these others take place, but there is no G plus C reaction. Hydrazine in the presence of NaCl. Okay, so this is one of the Maxim Gilbert reactions. Hydrazine in the presence of NaCl opens up cytosine. Right? If NaCl were absent, it would open up cytosine and thymine. But in the presence of NaCl, it only opens up cytosine. Cleavage reaction in Maxim Gilbert method is used to cleave phosphodiester bond next to the modified nucleotide. This is done with the help of piperidine. Right? Hot piperidine is used to cleave the phosphodiester bond. Okay. Here we have a question which shows this figure pictures an autoradiograph of the sequencing gel of a DNA that was treated according to the Sanger method of DNA sequencing. Okay. What is the sequence of the template strand corresponding to bases 50 to 100? Okay. From here to here. So essentially we need to read off this method, which we need to read upwards. So we would go T, A, G, C, T, G, T, 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 C, C, T. And then here, this is probably a G, so we put that. Okay. T. Now, ideally, we should have gotten a band here in between these two. Right? But these two bands are very prominent, but we don't really see any other band here. But the spacing between these two bands suggests that there should be another base here. So we can add question mark. Right? T, A, T, T, G, T, T, A, T, and so on. So you can just read the sequencing gel here up to 100. But you have to remember that what the sequence gives us is the sequence of the complementary strand, which was synthesized using the template strand. What the question is asking for is the sequence of the template strand. So what you'll have to produce, remember this is the 5' end, this is going to read up to the 3' end. What we now have to find is the reverse complement of this sequence, which will give us the sequence of the template strand. Feel free to sit and zoom in this page if you want to. You do this as an exercise. Okay, just an exercise in reading the sequencing gel and finding the sequence of the template. Okay, so we discussed what DNA sequencing would look like. But you can imagine that we can have a similar procedure where we determine this primary structure of a protein. So we know the different levels of structure within the protein molecule, where the uppermost would be the quaternary structure, which is how the subunits of the protein associate. The tertiary structure is one level down, which is what the one complete protein chain structure would look like. You can have secondary structures, which is what the polypeptide chain adopts, but the sequence of amino acids within the polypeptide chain is called the primary structure of the protein. Right? And obviously it is this primary structure which informs how all of these other structures form. Right? And even a small alteration here can have a large effect on the final structure of the protein. And so there is large amount of interest in actually determining what the primary structure of a polypeptide chain would be. So we can determine that through techniques of protein sequencing or peptide sequencing, okay, which we'll discuss here. So how you prepare protein for sequencing? First, you have to determine the different number of polypeptide chains. There's a different number of subunits. Cleave the disulfide bonds and separate the different subunits. And then we have to sequence each polypeptide chain. Okay. And how you would sequence a chain, it's slightly more complicated than the DNA sequencing procedure, because in DNA sequencing we can benefit from the fact that there is this DNA replication machinery which is going to synthesize new DNA on the basis of template DNA. There is no protein replication machinery, right? So if we have a protein, we have to basically break it into its constituent parts and determine what the constituent parts are. So you can see what this is what a set of a chain of amino acids will look like, where we have a peptide bond. Okay, we have this is one amino acid residue, and at one end of the chain, which is called the N terminus, we have this free amino group. This is a primary amine group. The other end, the C terminal end, is involved in this peptide bond, followed by the second residue in this chain, followed by another peptide bond, okay, followed by the next residue, and so on. Okay, and at the other end of the chain, we'll have a free carboxyl terminus, which is called the C terminus, right? NNC. And what we can do is determine what is the sequence of these amino acid residues within this chain. And how this is done by something called end group labeling, where if this is the chain of amino acids, in the labeling procedure, a label is added specifically to the N terminal amino acid, right? Well, yes, it's usually the N terminal amino acid. We don't have any reliable method of C terminal labeling, so it's usually added to the N terminal amino acid, right? It's reacting with the free amino group at the N terminus, which is followed by a step where this first labeled amino acid is released from the chain. Okay, so then we can detect what is the identity of this moiety which has just been released from the chain. After this has been detected, we are left with the rest of the chain. And so what we can do is repeat the cycle where we again label, and now it attaches to what is now the most exposed amino acid at the N terminus, which will be the second amino acid. Same, we will release the second amino acid, detect its identity, and then continue this into a third round. We detect the identity of 3, 4, 5, 6, and so on. So we can sequentially basically label the N terminal residues of the amino acid chain and detect them. There are two ways in which you can label the N group at the N terminus of proteins. These are all N term. One is by labeling it with dancyl chloride, okay, which is basically abbreviation of this molecule, 1-dimethylamino-naphthalene 5-sulfonyl chloride. And when this reacts with primary amines, it will basically attach to the N terminus of the polypeptide chain. And upon acid hydrolysis, what is going to happen is the polypeptide chain will be broken at each peptide bond. But only the residue that was there exposed at the N terminus of the chain will be labeled with dancyl chloride. Dancyl chloride is a fluorophore which has very intense yellow fluorescence, and it is only the N terminal residue which will display this N terminal fluorescence. So this only labels the one residue which is there at the very end. Okay, but if we want to label residues sequentially, we have to use something called Edmond degradation, okay, which is by far the most common method of peptide sequencing, which is very useful for short stretches of amino acids. Edmond degradation, we need a chemical called phenylisothiocyanate. It's also called Edmonds reagent. And what Edmonds reagent does is react with primary amines. Okay, you require a primary amine. Okay. And this will form adducts. Okay. And when we react this with trifluoroacetic acid, we will cleave only the N terminal residue, okay, and leave the rest of the chain intact. So the N terminal residue will be liberated as a PTH derivative of the amino acid, and then we can run this in reverse phase HPLC, okay, and detect the identity of this amino acid. Okay. So again, what N group labeling looks like. So this is dancyl chloride, which is a fluorophore. When it reacts with a polypeptide, okay, what's going to happen is that it will label this primary amine over here, and in the presence of acid and water, the polypeptide will undergo acid hydrolysis at each of these peptide bonds, and we'll end up with a large number of free amino acids, but the N terminal amino acid will be labeled. It will be a dancyl amino acid, which is fluorescent. And we can detect the nature of this amino acid. Edmond degradation, on the other hand, will look something like this, where this is our Edmond reagent, phenylisothiocyanate, is going to react with this primary amine. And this is the PTC polypeptide, where we have this adduct which is formed at the first amino acid. In the presence of trifluoroacetic acid, we are going to have cleavage here, okay, after the N-terminal amino acid, where the rest of the chain is left intact. So we will have the original polypeptide less its N-terminal residue. Now we will get this derivative of the N-terminal residue, which is called the PTH amino acid, okay, because it has this adduct added to it. And what we can do is then detect this through HPLC. Whereas the rest of the polypeptide chain can go back and react with PITC again to release the next residue. Okay? And in this way, we can sequentially release the N-terminal residues and determine the sequence of this polypeptide. So PTH amino acids can be separated through HPLC. Okay, you can see that different amino acids will give peaks at different positions in this HPLC run. These DPU, DTPU, PEC, these are all common byproducts of the Edman degradation reaction. But if I now run an unknown amino acid in HPLC and I get a peak, say, over here. Okay, this is my unknown amino acid run. And when I compare that with the standard, I can then determine what the identity of this unknown amino acid is, just based on its retention time. So I know that if I'm getting a peak here, it matches the retention time of tyrosine. And so my residue was most likely a tyrosine. Then we can repeat this for each residue. So yeah, elution profile of peptide hydrolysate. We're getting a peak here, which corresponds to the phenylalanine peak in the standard, which is how we can determine that this unknown peptide A, or the unknown amino acid A in this PTH adduct is probably phenylalanine. And in this way, we can determine one by one the sequence of the protein from its N-terminus. Now, Edman degradation works well on short stretches of amino acids. And obviously, a polypeptide chain will be quite a long sequence. So we can break the polypeptide chain into shorter regions by using things called endopeptidases. Right? What this is going to do is basically cut your polypeptide chain at various points. And then you can sequence each of these chains using Edman degradation, and then put all the sequences back together to form your final peptide sequence. Now, we would rather use specific endopeptidases because then we will know where they will cut in the polypeptide sequence. For example, trypsin, right, is an enzyme that is highly specific because it will cleave the peptide bond, but it will cleave it specifically after a positively charged residue. So if we have an arginine or a lysine, trypsin will cleave the polypeptide bond following these, right, as long as the sequence this peptide over here is not a proline. Okay, so if this can be arginine or lysine, this cannot be proline. Then this bond will be cleaved by trypsin. Okay. Similarly, chymotrypsin will cleave after a bulky hydrophobic residue such as phenylalanine, tryptophan, or tyrosine. Okay, and so if we digest our polypeptide chain with one of these endopeptidases, for which we know the specificity, it will make it much easier for us to piece together our sequenced fragments later. Right. So the amino acid sequence of a polypeptide chain is determined by comparing fragments, or comparing the sequences of two sets of mutually overlapping peptide fragments. So here we have a polypeptide chain, and one set of fragmentation they've done is digestion by trypsin. Like we just discussed, trypsin will cleave the polypeptide chain after lysine or arginine, right, with any positively charged residue. So we have lysine, so trypsin will cleave here. Trypsin will also cleave here after the arginine. So the fragments we'll get are this to the arginine, and then this, starting in from the cysteine. They've also done a secondary method of cleavage called CNBR, cyanogen bromide. Cyanogen bromide will cleave the polypeptide chain after methionine. Okay? And so when we use CNBR on this peptide, we will get a different set of fragments. Right? So we'll get after this methionine, this, and this. And since the fragments are overlapping with each other, when we compare these two sequences, we can then recreate the full peptide sequence. Okay? Now, these sequencing techniques have become slightly obsolete with the advent of mass spectrometry, which allows you to sequence small peptides with a lot of accuracy. So this, for example, is how one might sequence this 14 residue peptide. What happens in mass spec is we get these series of ions, okay, called the B and Y series ions. And depending on their mass by charge ratio, you can basically do an addition series of each peptide mass, okay, and fit it into the differences between each ion weight, each M by Z. And you can do that to determine the polypeptide sequence. So now that we've understood the basics of peptide sequencing, we can do a few questions. First one is identify the unknown hexapeptide from the following data. Okay, so what do we know? So first, there are 6 residues in this hexapeptide. Complete hydrolysis gives equimolar amounts of arginine, glutamate, lysine, phenylalanine, proline, and tyrosine, which means that our 6 amino acids are these 6. Right? We don't have any repetition of amino acids. We have these 6 unique amino acids in our hexapeptide. Edmond degradation of the hexapeptide gives labeled phenylalanine. Now, we know in Edmond degradation, the PITC will attack the primary amine which is present on the N-terminal residue. So if we perform one round of Edmond degradation, we will identify the residue which is present at the N-terminus of the protein, which means that this hexapeptide must start with phenylalanine. Right? So phenylalanine must be the residue which is present at the N-terminus. Chymotrypsin cleavage gives a tetrapeptide, phenylalanine, and glutamate. Okay, if we remember, what is the specificity of chymotrypsin? Chymotrypsin cleaves after bulky hydrophobic residues such as phenylalanine, tryptophan, tyrosine, which means chymotrypsin must have cleaved after the phenylalanine. It is then given a tetrapeptide and then it is given glutamate, okay, which means that glutamate is the C-terminal residue of this hexapeptide because it is liberated after this hexapeptide. But it also means that whatever the 4th residue in this tetrapeptide is, must also be a bulky aromatic residue. Now, when we look at the composition of the hexapeptide, the only other site for chymotrypsin cleavage is the tyrosine. So the residue before glutamate must be tyrosine. Right? Because only then will chymotrypsin cleave over here. Okay. Edmond degradation of the tetrapeptide gives labeled lysine. So if we take this tetrapeptide, we know it ends with tyrosine, which means tyrosine is on the C-terminal end of the tetrapeptide. But when we perform Edmond degradation, what we will get to know is the identity of the N-terminal residue, which is lysine. trypsin cleavage gives 3 dipeptides. Okay. And Edmond degradation, the dipeptides gives labeled phenylalanine, proline, and tyrosine. Okay. Trypsin cleavage gives 3 dipeptides. So obviously trypsin will cleave after this lysine, right? Cleave here. So the one dipeptide will be phenylalanine lysine. Okay. Edmond degradation of one of the dipeptides gives tyrosine. So this is the other dipeptide. Okay, with tyrosine and glutamate. Since trypsin is cleaving before the tyrosine, the amino acid present here must be positively charged. So this must be arginine. And therefore, the amino acid over here following lysine must be proline. Okay, which is how the Edmond degradation will give labeled proline, because it's the N-terminus amino acid of this dipeptide. So just to recap, the peptide sequence of this will be phenylalanine, lysine, proline, arginine, tyrosine, and glutamate. Right? C. So this is the hexapeptide, which will give this data in the different degradation reactions. So thus, by using a combination of different reactions, it's possible to find out the sequence of amino acids within your peptide. Okay. You're planning to chemically sequence a novel protein and perform N-terminal and C-terminal N-group experiments. You can identify a single C-terminal residue, but do not detect any N-terminal N-group labeling. But you know all of your reagents are working perfectly. So what is the possible explanation? Okay. So to do this, we have to understand what is actually getting labeled in N-terminal sequencing. Right? So in Edmond's reagent, PITC is going to react with the primary amine group of your N-terminal reagent. If it is not reacting, that means that the primary amine group is not available for reaction, right? So that residue has been modified, which is why we are not getting any reaction from the N-terminal residue. And these types of protein modifications do happen. Often in eukaryotic proteins, you will find things like pyroglutamate, okay, where that amine group has been modified and as a result is not available for N-terminal sequencing. Okay. Which of the following is correct about Edmond degradation? Treatment of protein at pH 3 with PITC is correct. Treatment with anhydrous amino acid results in the cleavage of the C-terminal residue. This is not correct. Edmond degradation is required to determine the sequence of the protein. So, I mean, it's not required. Okay. Just give me a sec. But PITC will react with the amine group under mildly alkaline conditions, so this is not correct. So the only correct option here, or the most correct option of these three rather, is that Edmond degradation is required to determine the sequence of the protein. Okay. Another theoretical example, uh, while on expedition to the Amazon jungle, we've isolated a polypeptide. Okay, we think it's a growth hormone of a newly discovered species of giant spider. So we're very excited to determine its sequence. Unfortunately, a portable sequencer is not working, so now we have to resort to other methods. Nevertheless, we are using more primitive methods. We have obtained the following data. So first, they performed a reaction called hydrazinolysis. This will give us the C-terminal region, the C-terminal sequence. So what we know is that the C-terminus of this protein has valine. Dancer chloride treatment followed by acid hydrolysis. Remember, dancer chloride treatment will liberate all the amino acids but will label only the N-terminal amino acid. So we know that whatever amino acid we get through this will have to be the N-terminal amino acid. So dancer chloride treatment gives us the N-terminal amino acid, which is proline. Okay, now we have trypsin followed by Edmond degradation of the separated fragments. Now what we know is trypsin will cleave after arginine or lysine. Okay. And what we also know is this N-terminal and C-terminal residues of this amino acid sequence. So what we have to do now is piece together these fragments to recover amino acid sequence. Now, since we know that proline is the N-terminal sequence, that means that this must be the first of the Edmond degradation fragments. So we have proline, glycine, alanine, arginine. Okay, after which we would get a break. Okay. What we also know is that valine is the last amino acid, so this must be the last of the sequences. And it's been cleaved over here. Now the issue is that both of these fragments must lie in between these two terminal regions, but what we cannot determine is the sequence of these two fragments. Since we don't have a corresponding set of fragments which is overlapping with this, we cannot determine the sequence of these two fragments. So it could be serine, arginine, glycine, lysine. This is possible, but it is also possible that it is going to be glycine, lysine, serine, arginine. Okay, both of these possibilities exist. So within this information, we cannot determine the full polypeptide sequence. What we can determine is the sequence of the two ends, and we have some doubt as to what the sequence of this middle region of the polypeptide sequence is, since we have no way of confirming the sequence, the sequence in which these residues appear. So this is as much information as we can recover regarding the amino acid sequence. Okay, so that is the unit that we have on DNA sequencing and peptide sequencing. We have also discussed some techniques for analyzing DNA and RNA fragments, specifically southern and northern blots. So we'll go through that quickly. So a southern blot is named after a guy named Southern. Okay, that's how it got the name. And what it does is detect specific fragments of DNA. So what this makes use of is the fact that complementary DNA strands will bind to each other. So if I have this DNA strand, okay, in my reaction mixture, and I have this complementary DNA strand, which we'll call a probe, it will bind to this strand if it is present in my mixture. Okay, it will bind to the target. But first we have to have a condition in which this binding can take place. So the first step in a southern blot will be to separate a DNA, okay, because we want these strands so that we can determine other properties such as what is the size of the strand and so on. So first we'll separate this gel on an agarose gel, as we've discussed in early on in the course, maybe in week 2 or week 3 we discussed the preparation of agarose gels. Then we're going to transfer this DNA from the agarose gel onto a nitrocellulose membrane. Okay, it is now stuck on the surface of the nitrocellulose membrane, which means it is available for oligonucleotides to come and bind. So this transfer process I'll discuss in a few slides, but essentially in the buffer solution we are going to essentially perform electrophoresis, okay, where this will be transferred onto this nitrocellulose membrane. Then we're going to incubate nitrocellulose bound DNA with the specific probe as discussed, which is traditionally radioactively labeled, and then using an autoradiograph we will get the position of the DNA which is complementary to the 32p labeled probe. So we will be able to see the position of the labeled DNA only within this large mixture. Okay, and this whole process is called southern blot. Now obviously the process starts by the separation of DNA molecules. Again, something we discussed earlier on in the course, you feel free to go back and revise that. But basically an agarose gel is formed by repeating units of agarobiosis, and depending on the percentage of the gel, the pore size of the gel will be different, which will allow you to resolve DNA molecules of different sizes. And then load DNA solutions, and when we apply a voltage gradient, since DNA is negatively charged, it will move towards the positive electrode, and we can separate DNA based on their size. Okay. Now obviously depending on the size of the DNA that you want to look at, you should prepare a suitable percentage of agarose gel so that your DNA fragments of interest will be resolved properly. So if you want to look at really small fragments, you should be preparing a 2% agarose gel. Whereas if you're looking at very large DNA fragments, okay, you have to produce an agarose gel of lower percentage so that a DNA can be separated effectively through a larger pore. Okay. Now, how do we transfer after we've run the gel? Now our DNA is stuck in the gel, so how do we transfer it to our filter? So if you have a gel, okay, if you have a very thin gel, okay, what you can do is place it between two glass strips, okay, and then add a wet filter paper which is soaked in this transfer buffer. Okay, this is sodium chloride, sodium carbonate transfer buffer. It's called SSC buffer. And then place a nitrocellulose membrane strip, which is where we want the DNA to transfer to, directly on top of the gel. Add more moist filter paper because we don't want this setup to dry out at this point. Okay. Then place a dry filter paper. You can apply some weight from above to make sure that it transfers properly. So that's good if you have a small gel. If you have a larger gel, you would have to use a setup more like this, where here's our agarose gel containing DNA. Okay. Above that you'll see nylon membrane. These are filter sheets. Okay. And you'll see 3 to 4 Wattman's 3 sheets soaked in transfer solution, and this is serving as a wick. So this is sitting in this reservoir of buffer. This is allowing the buffer through capillary action to travel up into the filter paper and soak the transfer region. Okay. Above the filter above the filter paper sheets, we have paper towels, glass plate, and a weight. Okay. So of this process, the transfer solution carries the DNA from the gel to the membrane, as the dry paper towels on top draw salt solution from the reservoir up. Okay, so through capillary action, it's been drawn up, and as the result, DNA is being transferred from the gel to the membrane. The DNA binds to the membrane on contact, after which we can remove the membrane. Then we will dry the membrane to make sure that the DNA is stuck to it. Dry and bake it under a vacuum so it's stuck very firmly. And then we can proceed for autoradiography. So if you look at the power of this technique, when you look at the full DNA, we see this large smear, okay, of genomic DNA. But when we perform a southern blot for a certain probe, here this is beta tubulin cDNA, and look at the autoradiogram, you can specifically see the presence of all the DNA fragments corresponding to beta tubulin. So what would we be using southern blots for? So just as a case study, we'll look at a case of cystic fibrosis. Cystic fibrosis arises as a result of deletion of a certain a certain phenylalanine F508 within this gene. Okay. So this is the wild type phenotype, where we have the coding strand which produces this phenylalanine 508. However, in a disease allele, these 3 bases are deleted. Okay. As a result, phenylalanine is absent, okay, in the amino acid product, okay, which gives rise to disease condition. And so we can actually produce DNA probes, okay, one which is for the wild type, one which is against del F508, and detect the presence of these 2 alleles in the patient DNA. Okay, so here we see probes oligo N and oligo delta F. Okay. So if this probe gives a result, right? So if this probe gives a result, okay, we know that there's a delta F allele within this individual. Well, this will tell us if there's a wild type allele. Okay. So if we look at this individual, we see a hybridization band in both the wild type and disease alleles. Okay, so this is a heterozygous for the del F allele. Whereas for this individual, there is no band in the wild type allele, but only a band in the delta F allele, which means this individual is homozygous for the del F508 allele. So this will be a cystic fibrosis sufferer. Okay. These are both heterozygous individuals. This is again a homozygous for del F. This will be another diseased individual. Now, if we look here, we see a band in the wild type allele, but no band in the disease allele. So these 3 individuals are homozygous for the wild type allele. Okay. And thus, you can see we can produce a full pedigree map of this family and track the progression of the disease allele by using a southern blot. Okay. Now, southern blots are used to observe DNA, so we can use it as we just saw to look at changes at DNA level. But we can use a very similar procedure called northern, okay, northern blot, which will look at RNA. So the same way in southern, as we use DNA probes to detect DNA, so that would start with the isolation of genomic DNA. If instead we start with the isolation of mRNA, you can detect specific mRNA molecules using again a DNA or an RNA probe, since it will still bind to a complementary oligonucleotide. That will give us information about the level of presence of these different RNA molecules, which is essentially going to tell us the expression level of different genes. Right? So northern blot will have a different use case to the southern blot. This will obviously start by the extraction of RNA, followed by the separation of RNA on an agarose gel, transfer to a membrane, cross-link, then hybridize with a labeled probe. The labeled probe will usually be a DNA fragment, which we will have to label with our radioactive phosphorus. Okay? And once it is hybridized, we can visualize in an autoradiogram. So if we just look at what a northern blotting technique would look like, obviously similar to southern blot. We have to first perform RNA extraction. We use chaotropic reagents to inhibit proteins like RNase, which could degrade the RNA that we're trying to extract. If we want to specifically look at mRNA, we can select it by poly A plus selection. So this will normally be done by using affinity chromatography, right? You can do this by affinity chromatography, where we run it through a column which will have an oligo DT region where our poly A tails of the mRNA will bind. Or you can do it by production of cDNA. Okay, it's basically a reverse transcriptase reaction. We will use oligo DT as the primer, and it will cause the production of a DNA strand which is complementary to our different mRNA molecules. Then we have to separate our RNA species using agarose gel electrophoresis, as we did for the DNA. Blot it onto a nylon membrane, immobilize it properly, prepare the radioactive probe, hybridize, and detect specific mRNA, followed by quantification. So quantification will be densitometry. So we look at basically the density of, or the intensity of the band. Okay. So what are the advantages of northern blot? It's relatively simple, high specificity. It does not need to be 100% homologous to the probe, right? So we can use sequences even with partial homology as a probe. We can detect size of mRNA transcripts. So this can help us look at things like alternate splicing, right? So if we're getting hits at different sizes for, say, a single probe, that could tell us that we might have alternately spliced mRNA molecules. We can store the plot for several years, and you can easily do quality checks within the process. Okay, so there are several advantages of the northern blot. Also some disadvantages. Primarily that mRNA is quite a delicate molecule. So during electrophoresis, we run the risk of degrading the mRNA, okay, which would affect the quality and the quantification of expression. As we're going to be using radioactivity, we'll probably be using formaldehyde in our gels and so on. These are hazardous chemicals which will pose a risk for both the workers and the environment. Sensitivity is not great compared to some other more advanced techniques such as RT-PCR, reverse transcriptase PCR, which is another method of quantifying, so-called qPCR. Another method of quantifying transcript levels. detection of multiple probes is difficult. You have to run multiple northern blots. Okay, and yes, so obviously there are some disadvantages, which is why a lot of people have shifted to techniques such as RT-PCR. But this is what northern blot hybridization data might look like. Okay? So here they've isolated total RNA from different parts of plants. So it's root, leaf, and flower. Okay, where and they're looking at the expression of different genes. Okay, of so here there's something called alpha coding gene. So this corresponds to all the genes which code for alpha tubulin within the plant. You can see it's expressed in the root, leaf, and flower because we getting blotting results for all of these. However, when we look at the genes specific expression of some of these genes, TUA1 is one of the genes which is in this mixture, but that is expressed only in the flower. Right? It's absent in the root and the leaf. Whereas TUA3 is present in all three, but it's expressed more in the leaf region. Okay. So this way we can obtain specific expression level data of various genes using northern blots. Another example of northern blot analysis. This time we're looking at different growth factor genes, so EGFR, EGF receptor, EGF, and TGF alpha. These are different, can be used as cancer markers. So, and what they're looking at is the expression of these in the normal pancreas and pancreatic cancer cells. So if we look at this EGFR blot, okay, we can see that the density of the band obtained in normal cells is far less than that in cancer cells. Same goes for EGF and TGF alpha. 7S, so this blot is basically a loading control. So this is a RNA used, an RNA probe is used here that will be present constitutively in all cells. And this basically goes to show that equal amounts of RNA have been loaded in all the wells, and that the differences in banded density that we see here are not a result of differential loading. Okay, it's actually a difference in the expression. It's not because of something that we have manipulated in the experiment. Okay. But you can see for these three markers, there's a marked overexpression of these genes in the cancer cells compared to the normal cell. So this can be used as a cancer marker. Okay, do some questions related to northern blotting. Okay, the smaller the DNA fragment, in electrophoresis, the smaller it is, the faster it will migrate. So it will move further. So the faster it will migrate during separation by electrophoresis. Okay. Purpose of the southern blot is to look for specific nucleotide sequences in DNA, yes. Determine how closely two organisms are related. This can be a conclusion that we draw from a southern blot. And to identify the size of the fragment that contains the sequence. So I would say prima facie the two data points that you can get from a southern blot are A and C. Okay. Steps performed in southern blot should be performed in the following order. So first we have to digest with the restriction enzyme. This gives us smaller pieces of DNA which will be easier to analyze. Then perform electrophoresis, stain with ethidium bromide to determine that electrophoresis is done properly, then hybridize with the radioactive probe, and then probe it on an X-ray film. So first we should have 3, 2, 4, 5, and then 1. This is correct. What is the first step in southern blotting? First step is treatment of the DNA to break it into fragments, okay, which will be easier to separate and run on the gel. If we have one big piece of DNA, this is going to get stuck in the well of our agarose gel, and we will not get any meaningful data from doing a southern blot on that. What is the purpose of treatment of DNA in the southern blotting? To break the DNA into fragments small enough to be carried out on the gel. What is the principal advantage of electroblotting in southern blotting? So the principal advantage is that it is much quicker, does not require acid or alkali treatment to fragment the DNA. What is the purpose of the agarose gel? The purpose of the agarose gel is to separate the DNA based on fragment size. What are the three principal steps of southern blotting? So, three principal steps are what should we say here? Separation, treatment, transfer, DNA staining gel for membrane treatment, DNA separation, DNA treatment, membrane binding, electrophoresis, DNA digestion, and membrane transfer. These three are probably the three principal steps of southern blotting. What is the purpose of the membrane in southern blotting? Purpose of the membrane is to bind the DNA fragments tightly so that we can visualize them properly. What are the advantages of using nylon-based membranes? Will be option A. They have high binding capacity, transfer in alkaline conditions, and can be reprobed multiple times. The following is not an application. So southern blotting looks at DNA level, so we cannot use it for RNA quantification. We can use it for the other three. How is the target DNA sequence visualized after hybridization with the probe after southern blotting? That we traditionally we look at radioactive emission. What is the role of the probe? The role of the probe is to detect and bind to target DNA sequence. What is the probe in southern blotting? The probe in southern blotting is a DNA or RNA molecule complementary to the target sequence, which will bind to our DNA. What is the solid support typically used in southern blotting? This is typically a nitrocellulose filter. What is the purpose of denaturing DNA fragments in the southern blot? So, since we have to hybridize our probe with the fragment should be single-stranded so that a probe can hybridize. So we should denature our fragments so that we can separate the two strands of DNA and our probe can actually bind to the DNA. How many bands would you observe on a northern blot of mature mRNA produced from a gene containing 4 exons and 3 introns, okay, assuming no tissue-specific alternative splicing? Okay. So, a northern blot of mature mRNA, which means a fully processed fragment containing 4 exons, 3 introns. So, during processing, we remove these 3 introns, but we are assuming no alternative splicing. Okay. Which means there is only one transcript that is available, which will give us one band on the northern blot. Okay. So this is a slightly longer question where we've been asked the molecular map of a normal allele of a disease-associated gene. Okay, this is a normal allele corresponding to a disease-associated gene, where they're given us positions of different elements of this gene. So you have a Tata box, translation initiation site, the stop site, these are the exons, these are the introns, and the EcoR1 restriction and endonuclease sites. In the diagrams representing southern, northern, and western blots, draw lines to represent the DNA, RNA, or protein bands that we observe in samples. Okay. Assume no alternative splicing and that your probes for southern and northern blots contain the entire mapped region. Okay. So we want to know what the molecular map of this healthy allele would be. So let's start with the southern blot, which is against DNA. Now, in a southern blot, the first step will be to restriction digest our DNA so that we break it into smaller parts. So what is going to happen is that our DNA will be broken up into smaller parts by assume here we're using EcoR1 since we've been given where the EcoR1 digestion sites are within this gene. Okay, so if we digest with EcoR1, the fragments we will get is this one long fragment, this short fragment, and this longer fragment. Okay, we will also get sort of these secondary fragments. Okay, and this whole fragment. Okay, these are all possibilities after EcoR1 digestion. Okay. Now they're given here a scale where this much is 1 kbp. Okay. So if we look at this, I'll assume that this distance is approximately the same as the distance between these two EcoR1 sites. Okay, so this is 1 kb. Okay. Which means this whole region will be, let's say, 3 kb. This will be 2 kb. This will again be 2 kb. Okay, the start to here, let's say, is 1 kb, and this to the stop is, let's say, 1 kb for ease of calculation. Now what we have to do is add up the DNA length that is present between the different EcoR1 sites, which will give us the different bands on our southern blot. We are assuming that our probe covers the full length of the molecule, so we can detect any part which will actually contain parts of this molecule. Okay. So this is 1 kb, this much is 1 kb, so this whole region is 2 kb. Okay. And then 1, 2, 3, 4, let's say this region is 4 kb. Okay, so let's start with this big fragment over here, this EcoR1 site to this EcoR1 site. This will be 4 plus 2 plus 2 plus 3. Okay, so 4 plus 2 plus 2 plus 3 will be 9 kb. So we will get one band here corresponding to this fragment. So this is 9. This EcoR1 to this EcoR1 site we said is 1 kb, which will give us a band here. This EcoR1 to this EcoR1 just eyeballing the size, it'll be 2 plus 1 plus 1 plus 2, 6. So these are the bands you would get from these sort of sequential DNA fragments within this gene. Now, of course, EcoR1 is not a perfect enzyme. It may not digest at every single site. And so we also have the possibility of these overlapping fragments. Right? So we can also get the 6 plus 1, 7 kb fragment. Okay, so we can also get this. We can also get this 9 plus 1, 10 kb fragment. Okay, and it is possible, okay, that we might get this full length gene, okay, which will be 9 plus 1 plus 6, 16 kb, which will be all the way up here. Okay. So these are all the possible sort of bands that we can get in our southern blot, okay, where we are looking at different DNA fragments that we are getting after EcoR1 digestion of this gene. Now let's move on to the northern blot. As we discussed, the northern blot, we're going to look at RNA. Right? We will assume that what we're going to look at is mature processed mRNA, which means the intron sequences from this gene will not be a part of that molecule. In our northern blot, we don't do enzymatic digestion. We just directly run the total mRNA on the gel. And so there's no question of having these restriction sites. And we are assuming there is no alternative splicing. So we are going to get one transcript variant for the northern blot. also what we need to look at, what is the region of the transcript, right? So in the transcript, we will not have intron 1, we will not have intron 2. So we will have this 1 kb region followed by the 3 kb region and this 1 kb region before the stop codon. Right? So the transcript will go from here to here. So we have 1, 3, 1, which will give us a total of 5 kb, right? Which will be the size of the sample. Okay. And I know that we have not discussed Western blots in this session, but most of you probably do have some idea of how Western blot works, where essentially we use an antibody to probe for the presence of its target protein. So a Western blot will look at protein, and since there's no alternative splicing, we will also get only one protein product. Now we know the RNA has a product of about 5 kb, and they're saying assume that 1 kb DNA encodes a 10 kDa protein. So if we have 5 kb transcript, we should get a 50 kDa protein, which should give us a single band over here. So this is what northern, southern, and western blots will look like for this one gene. Okay. Now we have another option here where this is the same gene. Okay, so I'll just go ahead and write our measurements again. This is 1, 3, 1, 2, 2, right? One, 2, this is, we said was 4. So these are the measurements that we just sort of eyeballed for the previous gene. This is the same gene. Okay, but this has a mutant allele. Okay. How do we expect these bands to come from a sample from a person who's homozygous for the mutant allele? Okay. So we already discussed what bands will be present in the wild type. Okay, so if we look at the DNA bands for the wild type, we have 10, we have 16, we have 9, right? We have 7, we had 6, and we had 1. Right, so this was our southern for DNA, our northern for RNA, and our western for protein. Okay. In RNA, we know that we get a 5 kb band here, and in the protein, we should get a 50 kDa band over here. So these are the wild type bands that we determined in our previous question. So what is the difference in the mutant? There's a nucleotide substitution in the ECOR1 site, which is producing an early stop codon in the mRNA. Okay. So in our mRNA transcript, okay, the transcript is going to stop over here instead of stopping over here. But this will have a secondary effect of knocking out this ECOR1 site. Okay. So since this is no longer has that 6 nucleotide recognition sequence of ECOR1, we will no longer get digestion at this site. So this is going to change the nature of the bands within the DNA sequence as well. Okay, so now if this equar1 site was not present, let's look at what the different digestion products are. So one is this equar1 to this equar1, which will be 4 plus 2 plus 2 plus 3, 3, 5, 9. So we will get this 9 kb band. Then we have 2 plus 1 plus 1 plus 2. This is the 6 kb band. And we have the full length, which is the 16 kb. Right? And because we have knocked out this equar1 site, we are not getting the sort of intermediate sizes that we were getting in the wild type. Okay. Now, obviously, since we've created an early stop codon, the size of the mRNA will also change. So instead of stopping here, the mRNA will stop here. Okay. So now the question is, what is the length of the new mRNA transcript that is produced in this reaction? Sorry, from this mutant allele. So we will lose intron 1, lose the introns in the mRNA. So the size of the mRNA transcript will be 1 plus 2. So that will be 3 kb. Because the mRNA transcript will stop here and will not translate this region in the mutant. So we see a significant decrease in the size of the RNA fragment. And lastly, since the size of the RNA fragment has reduced, we will see a corresponding decrease in the size of the protein. Since, again, assuming 1 kb DNA encodes 10 kb protein, if our RNA is now 3 kb, we should get a 30 kilodalton protein. Okay? And so we can now see the difference in the mutant and wild type alleles on both a DNA level, on an RNA level, and a protein level. Okay, so this is as much as I wanted to discuss this week. Again, if you have any queries, please do feel free to use the discussion forum to write in during the problem solving sessions and so on. Yeah, other than that, wish you a good evening and